Jayne and I have been working together for a few years now, and we wanted to add a little bit of pizzazz to the talk. And so we thought about, maybe a movie theme, and since Hangover 4 isn't coming out for a year or so, we decided to go with Star Wars.
Good choice.
So, bear with us. So a long time ago, in a galaxy far, far away.
Oh, wait, come on. That's not it. It was actually just a few light years ago in the basement of the Science Center. Is that correct? Yes. But it's easy to see how you confuse the two because cell reception is about the same in the basement as it is in deep, deep space.
Well, I got to look at. I spent hours working on this. Check that out. All right, a couple minutes, but so, anyways, the way this started was hello. Okay, so it was a few years ago, 2006, that a bunch of us got together, biology and chemistry faculty, in the basement of the Science Center. And we're trying to come up with a way to increase collaboration between the sciences. Chemistry and biology seemed like a perfect fit at the time. And so after a meeting, we decided to apply for, or we found something of interest to the people in the room. And we applied to the Merck AAAS grant, and we won a 3-year grant. And it's a prestigious grant. It wasn't a lot of money, but after 3 years and involving about 20 undergraduates, numerous publications, we decided to continue this. Jayne and I had spent a lot of time and other biology and chemistry faculty thinking that this was a good start. We need to build up this collaboration and make it bigger, wider, broader. So we came up with the UD SMART Center. UD SMART stands for the Supermolecular Applied Research and Technology. And the goal was to take advantage of a lot of the different expertise of the scientists in the variety of different fields. Also, we were hoping to use this center to help mentor new faculty, to give undergraduates and graduate students a holistic research experience. In other words, give them a chance to take something from the bench, chemistry bench, into the cell, whatever, into application. I had to add this to boldly go where no one has gone before, because I'm not really into Star Wars. I'm more of a Trekkie, so I had to do that.
And this is just to prove that we actually did get this grant, and also to call out the individuals who were equally important, and that is Mark Masthay and Mark Nielsen, who I hope are in the audience, and they were very instrumental in this grant as well. And also, I think Shawn is being a little too modest, so I'll say that when we went around the room and talked about what our current interests were, and Shawn said porphyrins, I said, that's it. We have to do porphyrins because I have been in love with porphyrins since I was in college. And my daughters rightly call me a nerd, but you'll see why porphyrins are so important. Not only are they responsible for evolution on the planet, but you'll see other reasons as well. So really, Shawn's work was the catalyst, and Shawn did the heavy lifting on writing the grant, but I also want to give the credit to the others involved, Mark Nielsen and Mark Masthay. So with that, I'm going to sit down and let Shawn talk for a while.
So what I wanted to talk about was I guess, a more serious subject, cancer. I think most of us have some experience with cancer. Either we know somebody who has had it, a friend, a family member. What we're focusing on now, we've looked at in the past different types of cancer. We're focusing now on lung cancer. And lung cancer causes about 1.56 million deaths annually worldwide. And you know, with lung cancer there are traditional treatment options. Surgery, radiotherapy, and chemotherapy. These are things that we're all pretty familiar with. And they've advanced over the last decades and, of course, have become pretty efficient, but they are not without their downsides. What we're focusing on is, I won't say a new area, but it's relatively new to, in particular, lung cancer. One wouldn't think that photodynamic therapy would be of much use to treating lung cancer. But in fact, in the last decade, it has become more of a method for treating certain types of lung cancer. The advantage that photodynamic therapy has over the other methods is that it's a more targeted type of therapy. And so, what is photodynamic therapy? Well, quite simply, what we do is we develop, in my lab, what we do is develop the drug, if you will, what we call the photosensitizer. And the idea behind this, as you know, in chemotherapy, what you're doing is you're giving a chemotherapeutic drug, which is, for lack of a better term, poison. And the idea is that because cancer cells are proliferating at a much faster rate than normal cells, they'll take up more of the chemotherapy drug and die before you do, hopefully. So with photodynamic therapy, we use a photosensitizer, which is essentially benign, which is something that can be broken down by the body and doesn't cause any cytotoxic effects. So the idea behind this is that if you've got a tumor site here, you would inject the photosensitizer. You wait some period of time for the photosensitizer to accumulate at the tumor site. Again, it is not active at this point. It's not doing anything to the cancer cells until you irradiate it with light. And then, of course, the hope is that you destroy the cancer cells. And, of course, this is a kind of a Jablonski diagram. The idea behind this is, and this thing is not lighting that up. Okay. You're right. That is a problem. Yeah. I was going to, if I stare at that, that's probably not good. So essentially, we take the photosensitizer in the ground state, and just by using, literally, a light bulb, we photoexcite it into the excited state, and then it goes through a number of different decay processes. Now, in and of itself, the photosensitizer isn't the chemotherapy agent. Like I said, without light, it doesn't do anything. But also, even in the excited state, it typically does not react with the cancer cells. What happens is, in the excited state, it will create reactive oxygen species. And of course, once you have reactive oxygen species, then it starts to react with the biomolecules in the cell and can lead to cell death. Now, a really good photosensitizer is porphyrin. That's really bad. Porphyrins are macrocycles, shown here. These are two different types of porphyrins. These are very common. They've been around for 100 years or more. The thing that's really interesting about these is that they have really strong absorption of light, and that's what a photosensitizer needs to be able to do. Also, they're found in nature. As Jayne mentioned earlier, they're very common in many living systems. So that being said, they seem like the perfect target. The problem, however, with most of these porphyrins is that these are completely insoluble in water. This is a tetraphenyl porphyrin, and it's like a rock in water. It won't dissolve at all. And of course, if you're trying to use this for physiological conditions, you're not going to have much luck. But there are a variety of porphyrins throughout nature. It's found in chlorophyll as an analog of the type of porphyrin. Of course, heme in hemoglobin is a type of porphyrin macrocycle. It's found in cytochromes. It has many different functions. It can transport oxygen. It can absorb light. It can be a catalyst, or it can undergo electron transport. So what we did in our lab was we tried to come up with a compound, a porphyrin, that would have certain properties that would make it a good photosensitizer. And so we had synthesized this many years ago. And then after talking with Jayne some years ago, she had found a certain type of porphyrin that worked very well in bacteria. And I thought, well, I think I can maybe improve on that. So what we did, and I'm not going to go into the details of the synthesis here, but essentially what we did was to take this very water-insoluble compound, and by putting methyl groups on these nitrogens, we create a cationic species. So this thing is extremely soluble in water, which is important, of course. The next step was to insert zinc. Now, there's some reasons for that, which I won't go into detail about, but one of the lesser reasons, I guess, is that I'm an inorganic chemist that works with metals, and to be quite honest, this thing is kind of ugly to me without a metal. So I chose zinc for certain reasons, and we inserted zinc. So this is the compound that we're working with. Jayne and her graduate student, Caitlin, came up with the name Zinc for this. That's not an IUPAC name, that's our own name.
It's a pet.
Yeah. So essentially what we do in my lab is that after we've made and characterized the compounds, we look to see, can we photocleave DNA? That's something simple that we can do in my laboratory. And so there's a lot more to it than this. But essentially what we do is we take our compound and plasmid DNA, and we irradiate it with light, and then we do gel electrophoresis, and those of you familiar with that, you know that you can determine if you've nicked the DNA or chopped it up into pieces by this method. And so that's kind of a preliminary study. So we synthesize it, we characterize it, and then we do these initial tests. And then we get involved with our collaborators, and in this case, what we did was we were working with Dr. Kango-Singh's lab, and we decided, well, obviously she works with fruit flies, and since no one really, there's not so much in the way of hoops that you have to jump through working with animals, no one really cares if a fruit fly dies. So it was a perfect model, and genetically it's very similar. The interesting thing, and I'll not go through this in detail, but what she did, and she came up with this method, was from the larval stage, she started incorporating Zinc into the food of the fruit flies. And the idea behind it was to see, is it toxic to the fruit flies? And does it get into cells of the fruit flies? And they did a number of studies, and they found that it was not toxic. And also what they found was that it does indeed get into the cells. And these are, I believe, the salivary glands. And you can see with the porphyrin-treated porphyrin treated, porphyrin eated, I guess would be the better one. But essentially, you can see that the Zinc actually can get into these cells. So the next step was to see, well, what does this do in terms of treating lung cancer? And so I started working with a guy at Wake Forest Medical School who actually works with lung cancer and does photodynamic therapy. And so he did some very simple studies. So essentially, he was looking at the A549 lung cancer cell line. And again, hard to see, but as you go across this axis here, this is the concentration of, we gave him both the zinc porphyrin and the one without the zinc. And as you can see, this is the viability of the cells. And it's important to note here, this is without any kind of light. Okay, so this is all done in the dark. And so when you get up to, well, approximately 100 to 150, or is that 250 micromolar, then it starts to have some cytotoxicity. The next thing he did was to do a similar study, only this time he would irradiate the cells with light and monitor their viability. And the interesting thing here is that what we're talking about in terms of concentration, so down around here you get, well, almost 100% killing, but these, I can't see that myself, I'm standing right here. This is 75 nanomolar, not micromolar, nanomolar. So this is trace amounts of the compound. And when you hit it, and so this is in no way cytotoxic without light, but you hit it with light and you get nearly 100% killing. It turns out that both the Zinc and the un-Zinc have a similar effect. So it looks like this work is progressing. We're working with him to try and do a little more in terms of these studies. But I'm going to turn it over to Jayne. You're not using this thing? Can I take this off?
Can you hear me with this? Yes, all right.
Yeah,
I'll just go with this.
I'm not going to wear this because it's very tough.
Well, that's fine. Don't worry about it. Oh, all right. Thank you. Are you done?
Yeah, that's it.
Okay.
Yeah.
All right. So what Shawn has done very nicely is tell us about a compound that he made that no one else in the world apparently has ever made before. And because of that, we have 2 patents. The first one was with myself and Tracy Collins, a Ph.D. student in my laboratory, and that was granted a 1.5 years ago. And then a year ago, we put in another patent on Shawn's zinc porphyrin, and we're waiting to hear back about that. So we've been very productive with undergrads and graduate students. And so what I want to talk about is, I'm a microbiologist, so I'm going to talk about microbes. So when we first started talking about porphyrins and what they might do, there had been a few little reports in the literature about using photodynamic therapy to kill microbes, okay? And so I thought, all right, we can try that as well. But just to set the record straight, of all the microbes on the planet, and believe me, there's orders and orders of magnitude more than people, the vast majority are beneficial to hosts, and in fact, 9 out of the 10 cells I'm wearing today are microbes, okay? So we are only 10% human if you want to look at it that way. But I'm going to only talk about those that are harmful to the host because that's what we worry about, right? We worry about the pathogens. And so we're going to be on this end of the spectrum. All right, so here's a brief history of treating infectious disease. All right, so in 2000 BC, it was here, eat this root, and good luck. And then we moved to a different sort of plan, which was that is heathen, so here, say this prayer. And then we fast forwarded to the era of Louis Pasteur and his colleagues, Robert Koch, and that was, where we discovered that microbes actually were the cause of disease, and we started to develop some very, very poisonous compounds, potions that might kill them, and we hoped and prayed again still that that worked. And then 1920, the advent of sulfa drugs, something a little bit better, and then we all know what happened, and that was Sir Alexander Fleming, the discovery of penicillin. So that's when we came up with the, here, take this pill, this penicillin, and it worked wonders for a very short period of time. So in the 50s, oops, bacteria had mutated. Here, take this different antibiotic, tetracycline. In the next 30, 40, 50 years, many more oopses, more and more powerful antibiotics. We ran through them very quickly because of overuse. I think you're all probably familiar with that. And then somebody declared at the turn of the millennium that the bacteria had won, and we're sort of back to here, eat this root. So there's some truth in that, and now we're in the race to find new antibiotics, and that's where I come in. So I just want to mention that the rest of this talk does have some disturbing images, promiscuous sex, suffering and social upheaval caused by microbes. And since we're running short on time, I'll skip the social upheaval part, but the rest I will cover. Okay, so revenge of the microbes, how bacterial resistance is undermining the antibiotic miracle. And it really was a miracle. The number of lives saved was tremendous. We took for granted that we could always outsmart the microbes, and we were wrong. And Abigail Salyers and Dixie Witt wrote in their book about what bacterial resistance was really about. And basically what's happening is that if you even had a single microbe that mutated in the gene that was resistant, made it resistant to, say, penicillin, tetracycline, whatever you're going to throw at it, it's going to have sex, unbridled sex. This is bacterial sex, so this is why the R rating. And so what's going to happen is that this mutated gene is going to be transferred to millions, billions of other neighboring bacteria, and pretty soon, if you treat with that antibiotic, yes, you'll kill a subpopulation, but those that have received the antibiotic gene are going to prosper and continue, and so there's the end, and the infection is going to continue. So what we have on our hands then is a very serious case where we have things like antibiotic-resistant tuberculosis strains. We have antibiotic-resistant strains of Staphylococcus aureus. You're all familiar with MRSA, stands for methicillin-resistant Staphylococcus aureus. So we've run out of antibiotics in some cases. Tens of thousands of deaths a year are attributed to antibiotic-resistant microbes. So it's a serious problem. President Obama has taken it seriously. I was going to talk about the seriousness of this particular organism and give us a little history lesson, but I think we're running out of time, except to say that during this 3 different pandemics, including today, we estimate 200 million deaths, okay? And we would have thought that's past tense, except for the fact, and of course it led to all kinds of crazy things like doctors wearing masks filled with chloroform because we didn't know what else to do, priests and the church being undermined, all kinds of upheaval. But what we really want to show is that the past is close, and it's often too close. So this was this weekend where we discovered in an ongoing investigation that research labs of the US government had been, not particularly taking care of their stocks and sending out stocks of that organism and other organisms. And we don't know if we don't really know the outcome of that. But one of the things countries like to do and governments like to do is fiddle with microbes and make them resistant to antibiotics and make them better biological weapons so that we hold the key and the others don't. So antibiotic resistance is a problem not only in the hospital, but in terms of biological warfare. Okay, so in addition to that, 60% of all infectious diseases of animals can also affect humans. And these kinds of diseases, called zoonotic diseases, animal infections, and the incidences of new and newly emerging infectious diseases of this type are on the rise. So as people move into cities and the environment is changing through climate change, we are seeing the rise of these kinds of infections. So what I study are biofilms, and I study organisms. Most organisms prefer, most bacteria I should say, prefer to live on a surface. Why? Because they're protected there. They're protected from predation, they're protected from dehydration. So what they do is a single cell attaches to a surface. They form these very complex structures. This is a mature biofilm. And so what I want you to remember just for the rest of the talk is that these are individual cells, but all of this other stuff, this hazy yellow stuff, is referred to as the extracellular polymeric substance or matrix, but we'll just call it slime, okay? So it's slimy and it's full of DNA, which is a very good glue, and it's full of polysaccharides, but mostly remember it's full of DNA, because you remember what Shawn said. Porphyrins can get into DNA molecules, and if you hit them with light, you break up the DNA structure. So our thinking was maybe we could do this with biofilms. We could disrupt the structure by treating them with the porphyrins. And again, the bacteria can go off and make new biofilms. So an organism that I work with is Pseudomonas aeruginosa. And it can form 2 out of the following kinds of biofilms. And so biofilms are good and they're bad and they're ugly depending upon. If you look at wastewater treatment facilities, the biofilms formed on the substrata are important to the degradation of toxic materials, carbonaceous materials, they denitrify. So it does a good thing there. They are bad in terms of forming biofilms in pipes. This can be a problem not only because they lead to corrosion of the pipe, but also because they can lead to disease. Legionnaire's disease was the result of biofilms of that bacterium forming within the pipes of the cooling and heating system within hospitals. Now, engineers just call this a correction factor, but they're actually very, very important molecular biological phenomena. And then, of course, we have the ugly, which is the biofilms that form on our teeth if we're not careful. So I know you'll all brush your teeth tonight. Okay, so as I said, I work with a bacterium that I've worked with ever since I was 25. It's Pseudomonas aeruginosa. There's sort of a slide missing here, so before I go to the next one, I just want to tell you a little bit about it. It's everywhere. It's in the soil. It's in the water. It's on your body. But it doesn't cause disease unless you happen to be a person who's immunocompromised, or you have like a cancer patient undergoing chemotherapy, or you're an AIDS patient, or you have cystic fibrosis. So, okay. So we're not going to get very far. That's the ugliness of Pseudomonas biofilms. So we took this porphyrin and we said, let's try it on biofilms. So what we did is we said, okay, we take light only. This is a confocal laser microscope slide showing you that this is what the biofilm looks like top down. This is it on the side view. So the cells that are green are alive. The cells that are red are dead. Okay, so we took light only. The cells are happy as can be. They're green, they're alive. If we pre-treat with the porphyrin and then hit it with light, they're all dead, which we confirmed by plating them. If we do a higher concentration of TMP, we have almost a complete peeling off of the biofilm. And here's what we noticed. If you do TMP only, the biofilm looks very different. It's opened up. And I forgot to mention, biofilms are about 1,000 times more resistant to antibiotics than the cells are if they're alone, swimming alone by themselves. So then what we did is we took it and said, okay, what happens if we add antibiotics? So we took tobramycin, which is a very well-known antibiotic treating Pseudomonas infections in people with cystic fibrosis, and the same kind of experiment where we did tobramycin only. So tobramycin is not being very effective against biofilms, which we know. Then we took tobramycin pre-treated with TMP, trying to open up the biofilm. So this is what's happening, is it's opening up the biofilm, and then treated it with tobramycin, and look what happens. Now, all of a sudden, the biofilm cells become sensitive to the antibiotic. Even more remarkable is we took an antibiotic that normally doesn't kill Pseudomonas, but with a pretreatment with TMP, this particular antibiotic, most of the cells are dead. So this is expanding not only the range of antibiotics, but the ability to kill. If we do, so what we're envisioning is that the TMP is going in, it's getting mixed into the DNA of the biofilm, that's the TMP. Then comes the antibiotic, it now can gain access to the cells where it couldn't before, and the biofilm is degraded. 2 last slides. We did this just to show with Zinc because Shawn said I could make a better one, and he did. And we found that as not typical, that under dark conditions, so those were light conditions, but under dark conditions, it turns out Zinc is extremely effective where no other porphyrin that we've ever tested has been. So this is Pseudomonas aeruginosa in the dark, and within 1 hour, we have killed 10 to the 8th cells per mil of Pseudomonas aeruginosa. Within 1 hour, we have killed 10 to the 7th cells of Staphylococcus aureus. And when we do the same thing with MRSA, the methicillin-resistant form of Staph aureus, we see the same thing. We get almost 100% killing within 1 hour, and by 2 hours, 100% killing.